Thakor from Johns Hopkins University and National University of Singapore. And I'll be assisted by Professor Bin He from Carnegie Mellon. And we'll have two other panelists and our ICANX challenger. Without further ado, I would like to invite Professor Bin He to introduce our speaker today. Professor
Thank you, Martin. It's a great pleasure for me to introduce our distinguished speaker, Professor Nitish Thakor, today. Professor Thakor is a professor of biomedical engineering, professor of electrical and computer engineering at the Johns Hopkins University. He also joined the National University of Singapore in 2012 and served as the founding director of the well-known Singapore Institute for Neurotechnology. Professor Thakor is a world leader and international pioneer in biomedical instrumentation and neuroengineering. And he has made a significant contribution and played a very important role to shape what the neuroengineering field is about as of today. Professor Thakor has published over 420 refereed journal papers, holds 16 US and international patents, and co-founded 3 active companies. He served previously as editor-in-chief of IEEE Transactions on Neurosystems and Rehabilitation Engineering. And currently, he is the editor-in-chief of Medical and Biological Engineering and Computing. Professor Thakor has won a number of awards, and I'm not going to read through all of them because it's going to take 10 minutes or so. So of them, most notable include IEEE EMBS Academic Career Award and EMBS Technical Activity Award, and among others. Professor Thakor is a fellow of multiple international societies, and including the IEEE, the American Institute of Medical and Biological Engineering, Biomedical Engineering Society, and the International Academy of Medical and Biological Engineering. Without further ado, Professor Thakor.
Hello, good morning and good evening. Bin, can you stop sharing? Okay, good. So I am really pleased to meet with all of you in maybe different parts of the world. So this is a very interesting and important opportunity to communicate ideas on the field of neuroengineering. And so I will start with perhaps telling you what is neuroscience in one slide or 2 slides with some nice videos, right? So when we think about neuroscience, we can think about neurons. These are brain cells. And how do those neurons work? Well, they produce what are called spikes, or they produce what are called action potentials, these little green spikes that you see. And below that is a video of hundreds of neurons working together. So you can see, this is our brain at work. Single neuron to maybe hundreds of neurons. And if we want to do neuroengineering, this is one of our big challenges, how to encode this and decode this information. Okay, but brain is much, much more than a neuron. Brain is this amazing organ, and you can see these 3-dimensional graphics showing how brain is organized across so many frontiers. I will stop here just to change the setting because maybe you cannot hear the audio, so I will see if I can give you the audio. I need to figure this out. Okay, okay, I'm not sure, so I'll just go back because this did not work.
Nitish, we can hear the audio.
Oh, you can hear the audio. Very
Yep.
good. Okay, thank you. I wasn't sure. Okay, excellent. So let's just go back to this, the whole idea that I was giving you about how the whole brain is organized. And this is a three-dimensional rendition of the brain using technique like diffusion tensor MRI. And you can see that brain is these 80 billion or 100 billion neurons wired together, 10 to the power 15 synapses. These complexity in MRI images, diffusion MRI, just incredible, isn't it? So I hope all of you are excited to think about brain and neuroscience. And in fact, I heard today that China has its own brain initiative launched just today. So it's really great that US has brain initiative, Europe has had its own initiative, and now China has its, right? So now let's move forward and define what is neuroengineering. Sorry, okay. So what is neuroengineering? So I will define it as that use of engineering to solve problems and make discoveries and translate these discovery through inventions. And I say that it can be both basic and also clinical. Basic means trying to understand this 80 billion brain population, or clinical means understanding, treating diseases of the brain, such as Alzheimer's and stroke and such. So I hope the basic idea is clear, but let's go into a bit more, in fact, quite a bit more detail. This is not easy. This is a Handbook of Neuroengineering that I'm editing. I hope it will be published later this year or early next year. And this, to try to cover the entire field of neuroengineering, I had to cover 15 sections and more than 100 chapters. And they include neural interface, neurosensors, devices, circuits, brain signals, and how do you analyze an information, imaging, the very popular topic of brain-machine interface, my own research nowadays on neuroprosthesis, neuromorphic engineering, so that's brain-inspired technologies, rehabilitation, cognitive at a higher level, cognitive engineering, neuromodulation, how do you modify brain function at cortical as well as peripheral level, and then clinical and translation. You see, it's this, in our lifetime, in last several years, this field has evolved so deeply that it's really an interesting and important time for us to be doing research in this area. Maybe let me break it into some topics, such as Neurochip, which is like a cellular level, constructing 3D constructs of the brain, or it's called organoids. Or neuro devices are like building electronic chips to implant in the brain. Of course, everyone is familiar with imaging of brain in many ways, and I mentioned prosthetics. And I will give a brief introduction of these 6 topics in 1 or 2 slides each. And then finally, my talk will focus on neuroprosthesis in a bit more detail, okay? So let's do our very quick journey of the field of neuroengineering. So let's start with something called Neurochip. What Neurochip is, in a microchips, microfluidics chips, you grow neurons. And here you see small axons growing, trying to find other neurons and forming network, right? So you can study, and this is 2D, but today's state of the art is 3D. And in this kind of a microchip, you can study basic science of neurons, myelination, or how these axons can grow, how they form neuromuscular junction between nerve cells and muscle cells, which is a fundamental basis to building neuroprosthesis. So you see, biologists and engineers work together at micro scale in this area. And now the state of the art, as I said, is even 3D, so that you tissue engineered brain in study disease models on a chip. Okay, let's give a very one slide, two slide synopsis of devices. Like these are usually bioelectronic devices. Now to do that, I should mention that devices, these chips are bidirectional. And when we say brain, but we really mean nervous system, which is cortical, spinal, peripheral, right? It's the whole nervous system. And this is like, I would call it hacking the nervous system, which means how do you go into the nervous system and create a bidirectional interface to the neurons or the nerves or the spinal cord, and takes designing these microchips, wireless circuit, integrated circuit, micro and nano sensors, and so on. This is the job of electrical engineers, biomedical engineers. Neuroimaging, of course, people are familiar with CT and MRI for clinical use, but researchers could use, in this case, our work on optical imaging to image brain tumors, so that you can localize brain tumor in a area where it's just starting to grow because it leads to changes in blood flow. And this is a concept, a design of a little microscope that goes into very fine detail, as you see, lots of nice color. But what you see here is that brain tumor produces changes in neurons. This is in green, and changes in blood vessels. This is in blue and red, and then circulation and oxygenation, right? So brain imaging can help us understand neurons as well as clinical problems. Now, let's go to neural interface, because of course we must interface to the nervous system in my work in neuroprosthesis. Well, let me show you a video, an example. Here is a person who has nerve injury. He cannot move his arm. So how can we interface to his nerves so that he can move an artificial hand, which you see in this table, that it's opening and closing. So this interfacing to the nervous system is one of the problems. And you will find that most researchers, as we did, work with animal model, like in this case, like a rat, we interface to its nervous system so that we can make its leg move. Or in this case, monkeys, who we can interface so that the monkeys can open and close their hand, and even individual fingers. So if you look at this graphics, you see that neuroengineering research is done on bench, as we call it, where interfaces to the brain and the nerves, and also finally, of course, our goal is to go to humans. Neuromorphic engineering. Well, can we make brain-like chips? This is the graphics. Can we compute the code of the brain so that this is some equation? Can we combine these and control a robotic arm? So neuromorphic engineering is brain-inspired, but in silico. That means in computational, in electronics. Well, there have been some fantastic example. IBM and others have come out with these brain chips, which then can be used like brain-inspired camera. So this is a camera that can freeze and understand smallest of the movements that on the left side that you can't see, but this camera, which is vision, brain-inspired, can pick it up. Brain-inspired touch, so that this can be used in prosthesis. So in this case, we are modeling the receptors, sensors on the skin. So neuromorphic engineering takes inspiration from the nervous system and implements in silicon in brain-like computer, brain-like sensors, vision, and touch. By the way, if you just look at the size of this chip, it has only thousands of neurons, hundreds to thousands, not 80 billion. So we have a long way to go. Okay. And last, I will just give example is cognitive engineering. Can we understand the brain, the mind, and how it does complex tasks. And of course, that needs to bring together the mathematical, the experimental, technological, and perhaps final, I'm thinking like a cognitive helmet, sort of the brain inspired, something we would wear that will create a brain machine interface. And I'll just, as an example, in this case, somebody who's, wears a brain cap and using that brain signals flies. And, he's wearing a virtual reality cap, and you can study the brain at work while the person is doing virtual reality flying, right? So we can now study brain at work in a dynamic fashion. So I hope you got an introduction to neuroengineering. I started with neuroscience, and then I give you some example of neuroengineering. And now I will go deep in one topic, okay? So this way, things that we saw as a big overview, we will now focus, and we will focus on neuroprocesses. Well, so first I have to say, what is prosthesis? There's old history, if you go back, even now, this is on the left side, what you see is the processes. It's essentially a hook. Fortunately, now about what last 20 years, with incredible investment by governments and companies, dexterous human hands have come about. And where will we go? Well, let's watch a video. This is a video that I saw when I was a graduate student. This is from Star Wars, and a human-like artificial arm is given to Luke Skywalker. And, it's not only moving, but it's also feeling, right? So that means in the last 30, 40 years, the advancement have brought us to this point, okay? So what is neuroengineering here? Well, how will we control these processes are? Okay, so let's see. So let's again give an idea how to control our processes, right? So here is, in fact, this was one of our first amputee subjects many years ago. And she's just thinking about moving her fingers. In this case, we are picking up signals from her muscles. And of course, how does that happen? Brain thinks, nerves get the message that goes to the muscle, and we decode and then move the fingers. Okay, let's fast forward. Now at our applied physics lab and consortium, much more advanced work is happening, where this gentleman is now quite conveniently and easily able to move his arm. And, okay, let me see if, so he can operate open and hand and fingers, change his arm position. By the way, you see that the arm and the hand look so anthropomorphic, as opposed to here, this person is wearing a harness. If you see this range of electrodes, and I have to play this video, is freezing. That's interesting. Some reason the video is freezing, so I need to go back to the playing. Okay, that is too bad. Let's. Sorry, I apologize for that, but looks like my computer has frozen, which is so unexpected. Okay, so we will start again. So here, okay, this gentleman has, you see a large number of electrodes and electronics associated, but he is now controlling the arm by thinking. The messages relate to the nerves, which is related to the muscles, but now on the chest because he has no arm. And then that information is decoded to control the prosthetic arm. So, you see that in 20 years, the technologies have moved so well to build this kind of a brain-machine interface. And now, here and now, like what is the current state of the art? Well, again, this is work at Johns Hopkins and Applied Physics Lab and the consortium, where a person is wearing, is interfaced, if you notice on his head, 2 sets of microelectrodes that are connected to the brain. And in virtual reality, you see these gray are these microelectrodes interface into his brain. And then he's controlling 2 arms. You see the physical arms. These are the 2, what are called modular prosthetic limbs. And in virtual reality, those arms are grabbing objects. Now, of course, in one sense, this is amazing because this is decoding the brain's activity to control 2 arms. In other sense, there's a long way to go, right? Because right now he's not wearing both the arms, and a lot of the electronics is not implanted as if like a pacemaker. But you see how signals from the brain are decoded and you achieve bimanual control. So the field has advanced very rapidly, and now we have good example of brain-machine interface. And in fact, we should say that our goal, can this person go home? So I'll give a couple of examples. First of all, how we do this. Well, we do this work in virtual reality first, so that these fine dexterous manipulation of the hand and fingers. So you see that real and virtually decoded, these different colors show that our decoding algorithms can track hand and finger movements. So to do this research, we can work with physical arm or work in virtual reality. Other things, without going into details, now we need to use machine learning, deep learning algorithms so that we interpret this complexity. So let me stop here, right? Isn't this amazing? I mean, this is a convergence of robotics, virtual reality, and machine learning, right? Everything like this, these cutting edge fields have to come together for us to do this. And it's fantastic. And our graduate students work on these problems. Okay, let's going back to reality. Well, you still need to do this, bring these processes at home. So here, one of our subjects is wearing the arm. He's using it at his home in a kitchen. This is a similar work from EPFL. Here, the gentleman is wearing the processor's arm, but he's, of course, a lot of things he has mounted on his body, but now he's operating his hand and fingers, and he can write, he can even drive, right? So his processor's arm being controlled is suitable for activity of daily living. So these are, by the way, muscle control signals, and eventually we hope that this will be brain signals when we go into daily living. Okay, so with this, let's come to a more recent research, right? And, so far what I showed you is motor, which means that brain thinks, and with that, there is a movement. Now, of course, in this graphic, it doesn't look like prosthetic arm, but we can make ESkin and cover it up. But now we provide sensory feedback. It's surprising, it's a difficult problem, because now you have to encode a sense of touch and convey it back to the brain. And there are many interesting challenges there. So this is to do bidirectional. So now, in this case, first of all, we look at organization of the brain, where there is a phantom limb, right? The percept, because amputee doesn't have a limb, but the brain has that phantom control possible in its motor and sensory cortex. And if the limb has to be controlled, there's one direction is to make it move, and sensory, other direction is to get that phantom feeling back to the brain. So this is now a very active area of research where both bidirectional work is done as well as understanding or cognition. Okay, so let's take it one by one, right? So I'm now going to talk about sensory part of the brain-machine interface. So, this is one of my graduate students who did some wonderful work, but we had set up an experiment on a patient. You see this thing's a little needle that is touching the finger. And when that needle is touching, we had microelectrode in the nerve, and on the screen, you see those little spikes, or it's on the right side picture, it's a nerve response. So in this example, you can see that touch receptors can encode information that can go to the nerve. And so this is what we call intraneural recording. Okay, so you see the sense of touch, a robot gives a little sensory information, and all the information of touch, and we'll see later even pain is picked up. So how do we model this? All right, so let's look at this graphic. Well, on the left side, we see the prosthetic hand. It's covered by these ESkin or electronic skin. It has sensors in a flexible textile-like layer. Those sensors are modeled after receptors, sensors in the skin. And then the neuromorphic engineering, we need to do brain-inspired nerve-like response. Remember, I showed you how nerves produce spikes. Well, our chips, our sensor and chips do the same thing. So if the prosthetic hand touches these different objects, this round, oval, and sharp, you'll see that different spike patterns are produced. So now the neuromorphic skin is able to produce very similar signals to the ESkin and then human skin. Okay, we're making progress, right? We wanted to build the sensors and give information to the person. So, well, this is the skin. And now if you're touching, what the laptop shows are these colors in red and black. Those are all the receptors producing that neural activity that is being perceived by that phantom hand, the hand. And now we put it on a prosthetic hand, so this hand is moving with 2 fingers. So the sensors are mounted on the fingertip. And if it's opening, closing, looking at different objects, shape, the ESkin picks up these. And in fact, if you see this sharp object, it even reacts to it. So it feels not just touch and the pain. We are making good progress. Let's understand how amputees feel things. This is really remarkable and complicated. A person who is amputee does not have a hand and fingers, as I showed. They have a residual limb, but they have phantom sensations. So this blue dot, or here the purple dot, and if you see these colors, are placed where they have a feeling, if you touch there, or if you put small electric current, that they feel a phantom sensation at different parts in the hand, that projected phantom hand that is missing. And these different nerves, the median and ulnar nerve, carry that message, and they give that feeling. So we can now give the feeling about touch of a finger by stimulating that blue dot or purple dot location. So my students studied that amputee for a year, and these are very stable representations. So which means that it will be possible to give long-term sensory feedback. You can give electrical stimulation, you can give pressure, and so on. So if we give the sensation by those sensors, what about perception? Can we amputee perceive these things? Well, so how do we give that perception? So researchers across the world are doing different things. I give 3 examples. What I showed here on the right side is doing it non-invasively. These electronic circuit and neuromorphic give stimulation to the skin. There's some work in Switzerland and in US where they connect to the nerves. And then there's work in Chicago where they microstimulate directly to the brain. So what you see here is the graphics. In this case, their work is on monkeys, but now we can do microstimulation of the brain. So sensory feedback, that neuroengineering of sensation is peripheral to cortical. Okay, so of course, it's much easier to connect to the skin and much more difficult to connect to the brain. So now what kind of sensory feedback you can give? Different amputees have different feelings, different regions. So if you see this mapping of 3 different amputees, you see these color dots represent different things for them. It's different fingers, different perception of pressure, buzzing vibration, cold, electrical tingling. Because the nerves were damaged and they are rewired in this amputation. So the map is different. So that's why we call it sensory mapping. It's a different perception map, and we have to work with that. So again, let's go back to amputee. This was a former student of mine, George. He is a bilateral amputee, and he, as you see, is operating his prosthetic hand so well. He has a very good control over the fingertip. You can see that on the fingertip are these epidermis or artificial skin-like sensor. Inside is the neuromorphic algorithm that takes that sensory information and produces like nerve-like signals. And then he gets electrical stimulation to feel that, right? So you see the whole loop. But now in this case, he's looking at a sharp object and he will feel some pain and he will react. You can see that as soon as he touches that sharp object, that stimulus is high and he reacts back. So I think this is a nice demonstration to show that it's possible to provide sensation and perception to an amputee. So you remember the Luke Skywalker video? This is something similar. Okay, there are ongoing research like Case Western, they've interfaced directly into the nerve to provide the feedback to the patient. So this, if you can do invasive surgery, he's able to grab an object and make sure that the object doesn't fall. So they're in this case, an amputee, it helps them grasp better. This is work at University of Pittsburgh, where they show that they can touch individual fingertip and the amputee can tell you which finger they're touching.
What you're seeing as the trials take place is that as the researcher applies light pressure to the robotic fingers, those physical sensations are converted into electrical signals that are fed directly back into Nathan's brain.
Yeah, so you get the idea. What it's showing is that touch signals now are interfaced directly into the brain to provide the sensory information. Again, unfortunately, these are not microchips that are fully implanted, but I think in the next few years that will happen. Okay, now the last stop. So we started with touch and sensation. We went to perception and cognition, like what does brain think or see? So this is, now I'm getting into a little bit detailed science. So please follow me. How does brain move or perceive anything? Well, so if you know about the brain organization, it has areas of the brain, what's called somatosensory cortex, that is related to motor movement as well as sensory movement. So if we want to do brain-machine interface, this is the area that's most important, at least for direct interface. But that's not that simple. Brain works in a very complex manner. It has a motor system. It has a somatosensory system. In fact, areas of the brain that do multisensory integration, because, for example, we touch, we have proprioception for position, temperature, pressure, vibration. There are many, and of course, you also then integrate visual information. If an amputee needs to go and grab a hand, they also see. So that means there has to be multisensory integration as well. So how do you study these complex brain networks? So this requires a technique called graph theory, where you need to study the connectivity of different regions. So it's a little complicated, and I'll try to show this first of all, again, just to give you this motivation to create these networks. First of all, we will have to put these electrodes, EEG electrodes, on the scalp so that we can create a full brain map of the this amputee's functionality. And then, when he touches or moves, or if we stimulate, you can see that there is a somatosensory activation. But these brain maps are complex, so we then break it up. So are there different modules, like motor or sensory? That's like a high level way to break it up. Or another way to look at it is much more detailed, like are there nodes, like specific group of neurons are active? Again, I have to say that this is still very coarse, because EEG and the brain doesn't have the resolution that we can get if we go directly to the neuron, but it lets us study the whole networks. So this is now recent research we worked on is like this multisensory integration. Can we integrate this sort of sensory information from many parts of the brain, and of course with and without sensory stimulation? So without going into detail, at a high level, we find that these brain networks get formed. For example, in the beginning of the movement, there is a motor cortex, but to some level, it involves sensory and visual input. In later phases, these progress, different parts of the network increase, right? So that in the beginning, you need to get a sensory input of where you want to go. And in later phase, what do you want to do, right? So this is the goal of these brain network, to understand how motor, sensory, touch, visual work together, okay? It's a little getting complicated because nothing with brain is simple, okay? And then this is even more messy, right? What are these circles showing? Well, please study, look at this a little carefully. These are different parts of the brain. And the color red is for the motor. The blue are somatosensory, and green are multisensory integration. It's not easy for you to read these letters, but remember, these are different parts of the brain. And what we now are finding are these networks that are connecting, that motor, somatosensory are working together. And then they do integration, the green, when they have to work together. And these are two amputees who have a very different network. So, things are getting complicated. I'm sorry, but that's the way research is. Sometimes studying brain networks requires very complicated processing of and using of this network and graph theory to understand this. And these are three different amputees. Don't worry about the details, but each amputee, if you look at the green color, which is the most interesting, is how they are, processing multiple sensory information and how they are encoding it so that they have a good brain-machine interface. And we are employing good mathematics and computational techniques to do that. Well, let me convert that into a video and a movie. It's much easier. This is my collaboration with Technical Institute of Munich. One of my graduate students then worked on this. Can you study temperature? And here, cold, warm, hot, so this, then can the EEG pick it up, right? So that means that you take this temperature information. If it's mild, you have one response. If it's noxious, which means very cold or very hot, these are different responses. So our very recent work is to study these dynamic interaction of sensory information and form brain maps of this sensory signals. So I hope this video gives you an animation of what we are doing. Like currently, this paper is just recently accepted. So I'm giving you some of the latest information. So let me kind of summarize now. I gave you a journey. So So let's see, what did I describe? First of all, I described to you motor processes. It's about movement restoration. Then we talked about sensory processes, sensory information and restoration of touch. So sensory and motor are both important in neuroprocesses. Going ahead, let's go. You remember you saw this picture some time ago. To make it all work together is a whole complex system with research work that we are doing on multisensory integration, where we need to understand how brain combines this information from the periphery, like touch, even visual input, proprioception, meaning position, in an integrated manner, and bidirectional, which means computer to the brain, brain to the computer. So today, in this frontier of neuroprocesses, the research is going on many angles, and I gave you a little bit of example of sensation, perception, and cognition. So I hope you see the big picture. Okay, so now it's not, I gave a lot of example just for the upper limb, but this is similar activity. This is Stanislas Povich at ETH Zurich is working with lower limb, where can you provide touch information to the foot, and he feels it because his brain is picking up the tactile information from the touch, and they interface to the sciatic nerve in this case, but he's able to feel the touch on the foot by interfacing to the nerves. Or this is a work by Pepe Contreras at Houston, where EEG-based system is able to interface to the person who is paralyzed, but is moving and using that exoskeleton to affect his motion. So this is not direct and complete brain-machine. The brain-machine interface is only doing start and stop, but it still helps that exoskeleton has some input from the brain signals. Here, as the field moves forward, this is work by our host today, Professor Bin He. His volunteer is wearing an EEG cap, and they have also decoded this continuous neural signals to control this robotic arm, and it's able to track the movement. And what's really beautiful here is how quickly and smoothly it's responding. So it's very recent work published by his team. This is work by a group in various institutions, Stanford and Massachusetts General and so on. They have decoded speech. So if you have an interface to the brain, from that you can derive and get speech, meaning text signals. And it's quite remarkable how accurately and rapidly speech decoding is occurring. So these are the frontiers. You get this idea that the field of brain-machine interface has some significant accomplishments by people around the world. So I want to conclude my talk with my thoughts on some of the topics and future direction. Okay, so first of all, some of my messages that we need sensors for processes, touch sensors, vision, proprioception. So it's an important area, and it's a marriage of robotics, processes, and of course, neuroengineering. I presented to you the idea of perception, right? That not only we make these sensors and electronics, but we need to study the sensation, the behavior, and then bridge to neuroscience. We start with sensors, we connect to the body for perception, and then cognition to understand how brain processes the sensory information, how it integrates multiple sensory streams. The sensors has well-developed engineering, but Of course, there is a need for making microsensors that are skin-like. Perception is some neuroscience, some behavioral and psychophysics, and cognition is a lot of neuroscience of sensory integration, those networks and graph theory, so that we can put it all together. And then the bigger challenge, so how do you make it work in real life, right? So we are at a stage where we are doing research to understand these things. But can we make it all work together, right? Sensory, perception, cognition, so that's very intuitive and interacting. So this is an important flow from engineering to real life translation. So there are, if you want to be involved in research in this field, there are many topics to study. Tactile sensors. I give some example of touch and pain and one video about temperature, but this is still has a lot of research and development to do. In the area of perception, how do you give feedback? Is it vibration on a skin? Like when you touch a cell phone, you get a vibration. Is it electrocutaneous to give a little tiny current? But the bigger problem is to do it in a skin is very practical for all amputees, but not very specific. In an invasive, if you give it directly to the nerve or to the brain, it'll be very specific, but it requires surgery. And that makes it very complex. And then, of course, what is that perception? So you need psychophysics. And then for cognition, I gave you the EEG and graph theory example. But again, Professor Hay is an expert on, dipole, source localization, getting into the deep details of where all the activities is, but you can also use functional MRI. And then the bigger challenge is how do you do this for the whole brain? Let me take the next, and before I end, some of the thoughts on how do you translate this research? So let's learn from these famous scientists, right? Bohr is a famous physicist. Edison was a great inventor. Pasteur developed really great biological processes leading to drugs that we use now. So what did they tell us? It's something you can learn, what's called Pasteur's quadrant. What it says that the research we do can be fundamental, which is like fundamental work in physics, or it can be use-inspired, right? Practical, like what Edison did. Or we can combine the two, which means in a basic research, we need to understand how brain works. So those of you who are interested in neuroscience, fundamental, how a brain works, that's pure research. And there's some of you are engineers. I want to build a prosthetic arm. Then it's improve the technology. But actually, in reality, taking the inspiration from Pasteur and so on, we must combine that basic understanding with technology to do what's called use-inspired research. So I hope the topic of neuroprosthetics tell you that we have prostheses that are practical as a technology that is advanced. And we are trying to understand that brain and neurons, how they encode to combine all of that. So I hope this is inspirational for all of you as a way to think about this basic and applied research. Okay, so to conclude, let's go to biomedical engineering. Biomedical engineering has done so many amazing things, but for our purposes, technologies like deep brain stimulation, cochlear implant are here. And they are fully implanted and treat disease like Parkinson or deficits like hearing. So I hope that these neuroprosthetics, I'm not saying I hope, I believe in coming years, it'll be a fully implantable solution to brain-machine interface in prosthetics. There are many successful areas: cochlear implant, deep brain stimulation over the last decade, fully come from research to practice retinal processes. And today I presented neuroprocesses. There's a lot of active research in periphery, phrenic nerve, bladder, treating diabetic neuropathy. These are just some examples. Again, I want to emphasize that it's not only brain neurotechnology, it's nerves, spinal cord, so on. Okay. This field, what is remarkable, it looks so esoteric, but actually there's a lot of technological growth. There are some economic numbers, a lot of details here, but it's growing. Neuroengineering, neurotechnology market is growing quite rapidly into billions of dollars around the worldwide. Even a topic like neuroprosthetics is seen to be a very high growth area. Neuromodulation is very clinically relevant to treat diseases like Parkinson's, and neurorehabilitation again relates to this kind of a stroke and other rehabilitation applications. So this is not pure research. This is very applied and useful to industry. And everybody gets that information because you hear about Elon Musk and Neuralink and who's building a brain-machine interface, and it gets everyone excited, young people, because they hear about these famous people. Google partnered with GSK, drug company, to create a company called Galvani. I mean, these are famous large companies, but most of the exciting research is happening in small companies. So there is a industry and job market for people in this field. It's not pure academic research. And so I think neuroengineering is revolutionizing medicine. It's each of these clinical areas being treated. I hope today I told you a lot about neuroprocesses to give you some of the details of this whole topic of brain-machine interface and how it controls the processes. It's both basic and applied. And I hope you are excited about this. In the beginning, we mentioned BRAIN initiative. Well, United States has been running the so-called BRAIN initiative for last many years. And this BRAIN initiative has many topics: large-scale recording and modulation of the nervous system. Remember, I mean, there are 80 billion neurons. So, we are today, maybe they're pushing the frontiers to say thousands of neurons. So even then, it's a very challenging work. The brain imaging, of course, there are CT and MRI scanners, but high resolution cellular level imaging is another frontier. Today, I did talk about tools for brain stimulation, but doing it non-invasively, electrical, magnetic, ultrasound. These are, again, important frontiers. How do you target to individual cells, not the whole brain, get down to single cell specificity? And then how do you study brain circuits? So this is the left side, the blue work. These are research frontier. On the right side are, how do you process that, right? So one side of it is, of course, the basic science, what you see in the bottom, theories, models, complex data. So there is a big role for data science. There are engineers and companies can work on technologies for all of that. And then since we are interfacing to the brain, it's really important to think about ethics. Like, are we doing it in an ethical manner in the best interest of the subject? So the BRAIN initiative has so many different examples and angles to it. So, well, if it's that important, can there be a Nobel Prize? Well, all right, let's see the Nobel Prize. These are Nobel Prizes in neuroscience. So let's look at them, okay? I mean, some of them are very old and some of them are new. And of course, they are fundamental basic neuroscience discovery, too many, but Hubel and Wiesel on the lower right side, they discovered how visual system processes information. That's why they got Nobel Prize. But they use microelectrodes and decoded spikes. More recently, Neher and Sekman, they studied single ion channels of neurons and cells, but they developed a technology called patch clamp. So you see what I'm saying is that, of course, neuroscience is fundamental discovery for which people get Nobel Prize, but in almost they've developed some technology. In those days, they didn't call it neuroengineering, but that technology enabled, like patch clamp required electrodes and certain type of circuits that made it happen to record from single ion channel. There are so many such examples, but everywhere I find that technology has played a role. And I hope that in future, this technology will be even more appreciated in more recent, for example, in 1979, computer-assisted tomography was given a Nobel Prize to Cormack and Hounsfeld. That is definitely bioengineering and neuroengineering. Other one is magnetic resonance imaging to Lauterbur and Mansfield. And then this graphene as a technology and fluorescence microscopy as a technology. So these are fundamental science, but now fundamental technology that has also given Nobel Prize. So you wonder whether this topic of neuroengineering will have a Nobel future. Now, I don't just mean Nobel Prize, but it will be Nobel as in we help human beings. You know, this gentleman, his video I showed you before, he's using the prosthetic arms to play piano. You know, it's so wonderful to see this progress, okay? So I hope I gave you a journey into a very noble topic or noble future. I acknowledge a lot of students and team here at Johns Hopkins, some of the company I worked with, Singapore, a number of people worked with me. And I want to conclude that brain is amazing. It has sensation, perception, cognition, and our work, they all come together. So I say, well, thank you for this kind invitation to work with us, listen to me, enjoy the journey with me in the field of neuroengineering. So I think I will stop there, and I hope I was able to properly show you all those wonderful videos and examples with which we can give you a view of what the field of neuroengineering is about. So thank you. And Martin and Bin, please take over. I'll be happy to discuss and take questions and so on.
All right. Thank you very much, Professor Thakor. Really amazing, great work. And I can attest that it's very, very noble to see that kind of progress where we can interface brain with technology. Amazing, very great. So we have a few questions coming from the audience. And the first question here is from Stein. And the question is, hi, Professor. What do you think about neuromorphic computing? Is it a good research direction?
Well, it's certainly among so many good topics. This is a good topic for those who are interested in modeling neurons and brain, like at a very specific level. For example, in our case, it's a touch sensor, but how does it encode the information? So that's what's called neuromorphic. Mm-hmm. But if you are a designer, then say, can I make a micro sensor that is like a receptor in the eye or on the skin? And if you're an IC chip designer, then say, can I make it into a circuit? So this is for the sensors. But I'm giving an example of that IBM chip. You can also make it's little computer, like hundreds of neurons, maybe thousands of neurons on a chip. So I would consider this as a very good applied direction. This field has started to mature where now some companies are, even Intel, like companies are researching neuromorphic computing because, let me give you an example. If you have vision, if you have a camera, it sees everything, the process, everything, face, eye, it's massive computing. In neuromorphic computing specializes and does it very rapidly. So it has certain advantages. So I would consider this as a good applied direction. It's just now entering industrial interest.
Very good. The next question came from Juan, and their question is, how does the pain sensor work? What is the principle behind it? Can it tell other kind of pain than a sharp tip? And can it maybe be caused by other sources?
Yeah, this is very good. And I'm glad you observed the videos and you were paying attention, because when we wrote that paper, the biggest reaction and interest was in the topic of pain. And, again, let's go back to the skin. It has receptors, as I said. But for pain, there are nerve endings, free nerve endings, that they pick up that signal. And then through very rapid fibers, that signals are relayed from that free nerve ending to the nerves into the brain. But after that, in the brain, that's a very complex process of where pain is perceived. In our work, actually, surprisingly, one of the biggest problem is what is called phantom limb pain. Because, I've used the word, right? Amputees have a phantom limb. They think that they have an arm. Their brain still thinks that there is an arm. But then because of that, they feel that the arm is missing and they feel what's called phantom limb pain. Nobody knows why and how that's occurring. So this is a very active clinical area of how to suppress that pain. Can you block the nerve message that goes from the periphery where the receptors are to the brain and stop the phantom limb pain? And yes, I agree that pain can be caused by many things, sharpness, which we showed, but also temperature, which we showed in a recent paper. But phantom limb pain is much more obscure and very difficult to understand. So yes, and it's a very clinically important area, and that's why physicians, clinicians, patients are interested in it.
Very, very good. I think in the panel, we'll get back to it because I still have some questions related to that topic. Our third question is about the brain and how it does its computation. So the question is, how does the brain do such complicated processing with so little power consumption? Is there any hope of bringing this to microelectronics? Or do we even understand the process?
Yeah, again, a really fantastic question. And I'm glad you all followed some of the details like neuromorphic and such. So brain consumes like about 35 watts of power, like a light bulb. So it's so remarkable. So yes, let's just be amazed by it. And if you look at computer chips, your cell phone, anything else, now having 100 billion transistors is not a big thing. The brain has 100 billion, 80 billion neurons, so it's not a big thing. Number of synapses are very high, 10 to the power 15, so that's still large, but we can think about a lot of networks too. So silicon computing chips are getting there in terms of numbers. Definitely, they are faster. The neurons work at millisecond spikes and 100 meters per second communication versus computer chips work at light speed. So that's not a problem. We are doing it. But it's the one area where there is a 10,000 factor problem is biology is 10,000 more efficient than current day silicon electronics. And that is why, again, this new interest in neuromorphic engineering to create the circuits, not just transistors, but the way the computing occurs so that it consumes low power. That is why they are trying to make it into microelectronic chips that are really low power. In fact, today, our cell phone or computers, one of the biggest limitations is heat and power that is generated. And I think, do we understand the process? It's just such a fundamental difference between biology and silicon-based industry that I think it's not a question of understanding, but more like how do you make silicon-based circuits that are low power? And there's a big gap. Of course, there is some research in bio-inspired circuits and so on that's very difficult to, for example, bridge neurons to silicon. So there's a big gap, and this is one of the grand challenges.
Very good. Thank you very much for the questions. I think now we will migrate to the panel. And our panelists today are Professor Xiang J. Duan from Peking University and Professor Jian Huang from Tianjin University. And we have our ESkin ex-challenger, Proxim Wang from Peking University. And I'd like to invite all members to join in the panel discussing the work on neuromorphic computing. All right, if we can get everybody on board to join Bin and I, okay, thank you. So welcome. Any comments from the panelists? Any questions?
Professor Thakor,
Yes.
Thank you for delivering such a great talk. I actually have two questions for you. First question is that when you talk about a lot of devices, and it seems that we are using a lot of biopotential measurement to do the sensing and to do the stimulation. So I'm wondering what will be the role of chemical sensing and the chemical stimulation in the whole process? Are there any way we can use effectively use chemicals in the future to do this kind of sensing and stimulation?
Yeah, well, very interesting and challenging question. The biology is really great. The engineering has not progressed as much. So if you think of, again, neuron and nervous system, we thought, and almost everything I presented was electrical, but actually, if you look at it, neurons and nervous system works through neurotransmitters. So that means it's dopamine, and its deficits leads to Parkinson's. It's glutamate, which excites the neurons, or GABA, which inhibits the neurons. So role of chemical is profound. Acetylcholine is important to connection between a nerve and a muscle cells. Now, therefore, the biology is very well understood. And in fact, there have been Nobel Prizes for that. Unfortunately, we as engineers have not, developed sufficient experimental and technological interfaces. For example, now you can use like carbon fiber, nanofibers even in using what is called oxidation reduction chemistry, you can sense dopamine, or other ways you can do glutamate and GABA. Very poor development. We can only barely get 1 or 2 probes in the brain, nervous system to understand chemistry. You can use MR, mass spectrometry, extremely difficult. So your question goes to the point that the technology in this field has not developed to the point where it is very useful in these kind of applied research like neuroprosthetics. And the one way I can advise young audience is, here is your opportunity. This is an area where it's less work is done, is an opportunity to do some great future work.
Okay, thank you. So one more question. So that we are using a lot of artificial intelligence network right now. I'm wondering whether we can use biological neural network as part of circuits or as part of electronics in the future, for example, to do the computing, right? We directly interface in the neural network and to read and to send data into the neural network and do the computing.
Yeah. So if you remember in the very beginning of my talk, I showed you neurons that were growing. And of course, that was a fast forward movie of axons going out and such. So it is possible to culture a network of neurons. There is also one of the great advances in the last 5-10 years is to work with these 3D organoids, as they're called, or even people call it mini brain. So it's a tiny few hundred micron nervous system. On the other side, there is microtechnology. So microprobes on cell culture, you can put electrode arrays and so on. Okay, so there are 2 fields, right? Neuroscience or biology, and there's microfabrication and even nanofabrication engineering. But when the 2 come together, the difficulty arises, because to sustain this biological network is very difficult. They don't live long. They grow in a very uncontrolled manner. And our micro neuroprobes are very invasive. So creating a symbiotic neurons to electrode has not been easy. Surprisingly, we can put microelectrode array directly in the brain and it tolerates. But at the biological biosilicon network, this has been going on for 20, 30 years. And my impression is that progress is very slow. There's some use for it, like some biological neural network in a dish or in a chip can be used for testing drugs. But to say that we have created true intelligent interface, I'm very pessimistic of the current situation. extremely leading edge work could be to work with these organoids, 3D mini brain, and study them using neural interface. But it'll be very, very basic research in that area.
Okay.
Professor Thakor, if I can follow up on that question about interfacing with traditional microelectronics. There are some advances more recently on liquid metals and phase transition in metals that can conform into very complex textures. Do you see those kind of materials playing a big role in helping pick up signals or deliver signals to the brain? And if so, what are some of the potential challenges of introducing these pliable conformal metals and so close to these neural networks?
Yeah, okay, let me expand a little bit. So what Martin is saying is liquid metal, which is what is called eutectic gallium, it's liquid at room temperature, it's flexible, compliant, and so you can make that into wires that are flexible, compliant. Now, you remember neuroscience started where Hodgkin and Huxley, Hubel and Wiesel, they put tungsten microelectrodes and things like that, or glass pipette. Now, everything is made in silicon because of silicon technology, so that these silicon microelectrode arrays are used to interface to the nervous system. The problem is that silicon is like needles, it's stiff. So what Martin is talking about is that can you make those probes flexible? So I would actually answer a little differently. In this regard, first of all, one direction is people are using carbon nanofibers and so on to make them so ultra thin that they're flexible as well as biocompatible, and they're so small that they won't cause injury. So that's sort of one direction. Now, coming back to flexible, therefore, these organic electronics leads to flexible, stretchable electronics, which is also important because nervous system, even our brain pulsates and our nerves are stretching and all that all the time. And then these liquid metals may be used for wiring because people forget they want sensors, they want to do electronics, but what about the wiring? And therefore these liquid metals have a role. I have not seen too much micro miniaturization of these things. So I think it's a bit more difficult that way, but they are used as interconnect. In fact, there was some effort for retinal processes. Remember, retinal process is like a chip that's going in the retina, and these liquid metals can be used as a connector from the chip to the rest of the electronics. So this is definitely very cutting edge work. And liquid metal is a good example. The organic electronics, certain stretchable, flexible materials that are being researched is very hot. In fact, there are these journals called soft matters and soft electronics and soft robotics. So, if you look up this soft robotics or soft electronics, you'll see this kind of research being presented.
Yeah, very, very good. Actually, just for the sake of the audience, we do have some work coming out of my lab where we show that you can actually start with a liquid metal, and then once you put it on the brain, actually it solidifies and forms a shape exactly as the texture of the brain. So yeah. I'm hoping to share that with the neuro community and get it going.
Very good, because that's what people forget, that there is a mismatch of what's called Young's modulus, right? Engineers know that Young's modulus gives you stress strain, and the problem is that current technology, Young's modulus and brain nerve Young modulus is off by 1,000 factors. So we really have work to do to make our sensors and electronics compliant. And that's why liquid metal is a good example, and certain type of polymers and nanofibers are a good example of trying to do that.
Very good.
This is a really great talk as always. I'm just wondering, you get into a lot of very exciting recent research in neuroprosthetics. I wonder, could you share your thought that this field has been so exciting, attracting so many researchers? What would be most important in order to move this field forward? So actually we are going to see that neural-controlled prosthetic limb widely used or adopted by the clinical community? And what would be the most important challenge we have to overcome? I would be very appreciative if you could share your thought.
Well, Ben, it's a big question, right? And let's say how challenges are, right? It's all started with neuroscience challenge, right? There's a lot to learn about brain, and we should continue to do that, even nervous system. Just now, Martin's question and some example, and Neuromorphic says we have a technology. And what I think the field of neuroprosthesis is doing is merging the two, right? So that the biology of nervous system and engineering of prosthesis and all kind of comes together. So I think that's good. And what it took for that to happen is, like in US at least, the war, the soldiers came with the amputated limb, and government said, well, our agency in US called DARPA said, let's put huge amount of money to make this happen. So I think that's the first thing. People have to see big clinical human need for governments to put the big money. What is now happening, and I hope it continues to happen in answer to your question, is the commercial interest and clinical interest, right? So that companies can then convert our research or even our prototype into products. And one of the problem often is that, like, a neuroprosthetics is so expensive and so few people, few thousand people, it's very difficult. And this is where economics can be a problem to scale up, scale down. So I think the answer to this question, I will divide it into research. I think it's in good shape. The brain initiatives everywhere are helping because it's giving a lot of funding. The development is in good shape because now engineers are working on neuroscience, neuroscientists use technology. The clinical needs are existing, but can you commercialize and translate? So there is hope, right? Deep brain stimulation is now a successful commercial product. Cochlear implant is a successful commercial product. Retinal prosthesis is getting there, even though it's expensive, even though it's advanced. So it'll take another, if you look at that time it took, another decade or so before a commercial caliber technologies will happen. And the cost will be reasonable enough for that to happen. And that, I think, we should not ignore, because a lot of people here are interested in R&D. And that's why towards the end of my talk, I did discuss some of the commercial aspect of it. And that's as much challenge as scientific challenge.
Thank you.
Very good. Professor Duan, do you have a question?
Yeah. I have a question. Professor Thakor just mentioned the neuroethics issue. So my question is, what do you think we should do now for, regarding this issue? Should we try to get some regulations, or is there any laws, things related? So what should we do now, or is it urgent now, or it is okay for the near future?
So it's a very important question, and it applies to all research. Of course, when brain is involved, people are even more sensitive. Mm-hmm. So, if I do a study in an animal model, in a rat or monkey, anybody does research, have to submit to animal care and use committees that regulate that. Are you doing research for good ethical purposes, not giving pain and suffering to the subject? Similarly, when I do human studies, any one of you, then you go to IRB, it's called Institutional Review Board and such, who follow similar protocol. Did the subject give consent and all of that? But now, once we start doing this kind of brain research, there's something a little higher, right? Like topic of pain or phantom limb. Makes us feel like you are interfacing to us as a human being, and that makes people a bit more sensitive to this line of research. So definitely a field called neuroethics is evolving. There are well-known ethicists who understand neuroscience and are providing guidance to the committees that fund this type of research. So I think the answer is that this should be in parallel, right? So that all research should be in parallel reviewed by social scientists and ethicists so that they approve the good procedures and good guidance.
Very good. Maybe because of time, we shift to our ICANX challenger, and we get him to ask a question or two.
Thanks for Professor Nitish excellent lecture. It's very fascinating. And I'm PhD candidate in Peking University. My name is Wang Puxing. I have some question. When decoding the brain activity, your statement is focused on EEG signal like alpha band in specific brain area are relevant with motor. Do you think that the single unit spike also contains message needs to be interpreted? And how can we combine them together and understand them?
Yeah, so very good question again. So EEG is at the whole brain level. Unfortunately, its resolution is very low. You can only put 60 for 128, 200, maybe that's it. It's some limit to that. Secondly, that signal is very low frequency, it's blurred. So therefore, yes, we can decompose into alpha, beta, mu band for brain machine, etc. Its advantage is it's non-invasive, so clinically it's applicable. But now you mentioned that what about single neuron, or neuroscientists call it units. And that's how the basic science 50 years ago started. And now where we are is not single neuron, but rather large population of neuron, because we realize that these thousands and even millions of neurons are working together. So now the microelectrode technologies, the optical fluorescence imaging technologies, optogenetic technologies, these are the ways that we can process, interface to thousands of neurons together. But these techniques are for basic science. So they can be done in studies in rodent and monkey models to understand how networks of neurons work together. And where in the brain-machine interface, I was only, because of limited time, give one example where that microelectrode array was implanted in the patient's brain and interface it to about 40, 50 neurons. But that's still good enough for movement. But we'll have to go to thousands and millions to really do very effective sensory motor integration. So I think there are always like two paths: non-invasive for clinical and ethical reasons and convenience, and invasive, like at this micro scale for more fundamental work. And then micro technologies will have to be implanted in humans, and that takes even longer. So both are useful, but if you're a basic scientist, more basic scientists will be working in single neuron and population neurons. And if you're an applied engineer, you will prefer to work on non-invasive technologies.
Very good. So, and the last question.
My last question is, my major is biomedical engineering, and it takes a lot of time to repeat when we research it. So does the field of brain-machine interface can further libel us from this kind of repetitive experiment, or could it bring us some new methodologies to this field?
Yeah, so, for example, when I was a student, it was electrical engineering. Maybe 50 years before that was only physics, right? Or some general engineering. So, education system also evolves. So after that, in 70s, 80s, even 60s, biomedical engineering evolved, and it's now, at least in US, it's the hottest field. The best students, the top programs are in biomedical engineering, because it integrates biology, medicine, engineering, computing, and people find it exciting that they are doing cutting edge work, but for human health. Now, within that, now is this neuroengineering as a more specialized topic. And, it's possible that I think it will itself be a specialized topic, just like, for example, nanotechnology has become a specialized topic, maybe even many departments. You definitely see the major academic centers have neuroengineering as a focus or thrust area. At Johns Hopkins, there are 7 thrust areas. One of them is neuroengineering, and I lead a training grant in that area as well. Industry, similarly, is now very consolidated. Major companies are doing brain imaging, implantable neuro devices, and now the emerging companies, the famous one because of people involved, Neuralink and so on, but many beautiful creative startups are doing neuroengineering. So both education and commercial development, if I was a young person, I think this is one of the hottest topics to be involved in. If you're mathematical, you do computational modeling. If you're an electrical engineer, you build circuits and chips. If you do nanoscience, you put nanoprobe, or material science like Martin's in liquid metals or whatever. If you are biomedical engineering, like Professor He, you can work on brain models and graphs. And if you are a MD, PhD, as in US, that is in education, then you say, well, I'll take this technology and implant it in human beings and treat a patient. So I encourage people to participate in this field. It's exciting, futuristic.
Very good. Now, for the sake of time, I would like to move on and say, Professor Nitish, it's a great pleasure. Now, if we had you in person, we'll present this to you, but because of the situation, please accept this virtual acknowledgement. We are very happy to have you as our speaker for volume 69. And as we move on, I would also like to remind everyone that we have in iConnect now, we have the Young Investigator Award, Young Scientist Award. So if you received your PhD in the last 10 years, kindly look into applying, sending your application by October 10th. These will be awarded every quarter, and we want to bring the best talent from across the world and to give them a platform to share their work. I also want to highlight our speakers for this month. We've had John Rogers from Northwestern, Masayoshi Esashi from Tohoku, and now Professor Nitish. And then next week, we will be having Professor Peng Xing from Peking University. Please continue to join us. And then next week, we are going to have Chenupati Jagadish as our co-moderator, and we'll have Professor Jin and Professor Hong as panelists, and our ex-challenger is going to be Mr. Bao from University of Technology in Sydney. And with that, I want to thank everyone. Thank you, the panelists. Thank you to our challenger. Thank you, Professor Nitish. Great talk. And thank you, everyone. And with that, we bring this to an end.
Thank you for your invitation. It was great to interact with you all.